Greetings everyone, my name is Vitor Favaretto Pinoti, and today I'm going to present my work entitled Cy1 Interacts with RNA Helicase 35, a splicing and transcription machinery associated DEAD-box RNA Helicase involved in plant growth control. The pistil is the female reproductive unit of the angiosperms, and its proper development and functioning directly impacts the yield of fruit and seed-based cultivars. Our group previously described the Cy1 gene, which controls the cell proliferation in the upper pistil of tobacco and Arabidopsis thaliana. However, in spite of a well-described phenotype, the molecular mechanism exerted by Cy1 during this process is still unknown. And to address this question, we performed a yeast-to-hybrid screening of a tobacco stigma and style cDNA library using Cy1 as bait, and we identified the RNA helicase 35, which belongs to the DEAD-box RNA helicase family of proteins that are known to be involved in the RNA metabolism as one of the interactors. We confirmed their association by in vitro pull-down and BYFC in plant as well. The RNA helicase 35 gene is conserved across the plant kingdom. However, there was no information about this gene in any plant species, which led us to characterize it along with Cy1. We then performed colocalization analysis with nuclear body markers, and we could observe that Cy1 and the helicase localize and interact with each other in nucleoli and splicing speckles, which are nuclear domains occupied by proteins involved in the splicing process. We then tested their association with the homologs of three proteins known to be involved in the splicing process, and we got positive results for all the 6 interactions, which does show their association with the splicing machinery. To gain further knowledge about the RNA helicase 35, we also performed its hybrid screening, as it was done for Cy1, and we identified the homologs of proteins involved in the RNA metabolism as well, the homolog of a transcription elongation regulator, and a set of transcription factors, including the notorious regulator of the cell proliferation, the TCP4. We then produced silencing and overexpression lines for the RNA helicase 35 gene, and by 40 days after germination, we could observe that each transgenic line presented 2 phenotypic groups, and in the case of the sinus lines, each line presented a group of plants that was similar to the control group and a group of plants where the plants were smaller than the plants in the control group. And the opposite was observed in the overexpression lines, where each line presented a group that again was similar to the control group and a group of plants where the plants were bigger than the control group. We quantified the mean leaf area of each of these groups, along with the RNA expression levels of the RNA helicase in each of these groups. And we observed that the reduction in leaf area in the sinus lines was correlated with a higher level of silencing within the extreme phenotype groups. And the opposite was observed for one of the overexpression lines, where the increase in the leaf area was correlated with a higher level of overexpression. Additionally, the extreme phenotype groups of the sinus lines also presented a reduction in the floral bud and petal length, along with a reduced stamen length and an increased stigma diameter, thus showing that the RNA expression levels of the RNA helicase 35 affect the final size of vegetative and reproductive organs. Altogether, we showed here that Cy1 interacts with the RNA helicase 35 and that both proteins are associated with the splicing machinery. Furthermore, the yeast-to-hybrid screening of the RNA helicase 35 indicates that it may be functioning as a co-transcriptional regulator by associating with transcription factors and transcription regulators. Additionally, we also observed that the loss and the gain of function phenotypes of the RNA helicase 35 resembled the gain and the loss of function phenotypes of the TCP4 pathway, respectively, which thus indicated that the RNA helicase 35 may be regulating the TCP4 pathway, which ended up regulating the cell proliferation, which also does provide us possible mechanism by which Cy1 may regulate the cell proliferation, which involves its association with the RNA helicase 35 and the impact of Cy1 the TCP4 pathway afterwards. I would like to thank my lab members, the institutions I've been working with, the funding agencies, and the organizers for the opportunity to present my work. Thank you very much.